master thesis presentation about my topic of research, abundance and activity of the gammaproteobacterial taxa UBA 10-353 from the EP to the bathypelagic waters of the global ocean. This topic has been under the supervision of Professor Gerhard Ernst and Dr. Chi Amano. Before diving into my topic of research, I want to talk a little bit about the biological carbon pump. The biological carbon pump is basically the phenomenon of transfer of fluxes of carbon from the atmosphere to the ocean. Basically, inorganic carbon enters the euphotic zone and is transformed into organic carbon through photosynthesis by the phytoplankton. This organic carbon is then transferred through the food chain and could be remineralized into the dark ocean and transformed into inorganic carbon before being ejected into the atmosphere. Some of this organic carbon, however, is sinking through fecal pellets of sinking particles to the seafloor and constitute the organic carbon burial. Since many years now, studies have shown that the microbial carbon loop has a very important role to play in this carbon cycling. And that's why we focused on UBA 10-353, which is a newly discovered gammaproteobacterial taxa close to Theoglobus and SOPO5. Theoglobus and SOPO5 are known to be key actors of the sulfur cycling, and very recent studies have shown that UBA could have this same impact, but on the carbon cycle. That's why my research topic is, and my question 53, a true actor of the carbon cycling in the open ocean. The aim of the study is to determine the abundance of the taxa UBA and its activity in the global ocean. On this map, you can see the different stations where my samples are coming from. So basically, we have one station in the Northern Atlantic coming from the Thor Geotrace cruise, one station from the Pacific coming from the Zona cruise, two stations in the Southern Ocean coming from a Moby cruise, and several stations in the subtropical Atlantic coming from the Archimedes 4 Mocha cruise. Here you can see the same map, but this time the colors refer to the substrate incorporated into sampling samples. So we have two stations incorporated with leucine and DIC, two stations with leucine only, and the station from the subtropical Atlantic are actually incorporated only with DIC. So, in order to study the abundance and the activity of UBA, we use two techniques combined, which is called microcardfish. Microcardfish is the combination of two techniques called cardfish and microautoradiography. Cardfish has been used to determine the abundance of UBA. It is also called fluorescence in situ hybridization. Basically, we hybridized the DNA of the bacteria with HRP probe, HRP meaning a horseradish peroxidase. It's an enzyme that catalyzes the oxidation of organic substrate by hydrogen peroxide. And this hybridization, once since done, we incubated it with the fluorescent aramide. And after some reaction, this fluorescence was attached to this probe and then to the DNA, meaning that the DNA of UBA was fluorescent under the microscope. In order to target the DNA of UBA, we use 7 different probes, 1 main probe, 2 competitors, and 4 helpers. And to target the bacterial material, we use UB338-1, which is known to target most of the bacteria. Then we use microautoradiography in order to catalyze and analyze the activity of UBA. So basically what we did is that we used the filters from the water was sampled through filters, and we did cardfish on these filters. And we used these filters from cardfish, and we transferred them on a slide glass covered with photographic emulsion. This photographic emulsion is very sensitive to the light. That's why we worked in a dark room. We put these slides in a tight light box for different exposure time, 4 days of exposure time for leucine and 3 weeks of exposure time for DIC. After this time of exposure, we developed the slides and we checked the result under the microscope, as you can see on the point 5 of this illustration. The point of combining these two techniques gives us the fluorescence of the fish signal coming from cardfish and the silver grain that allows us to distinguish the activity of the cell from the microautoradiography. After checking the slides under the microscope, we took 20 pictures for each sample and each depth, and we analyzed this picture with the actual vision software. You can see here the one picture coming from the software. So here it's the final picture, meaning the 3 channels has been all put together. The blue dots are coming from the DAPI staining, so it's prokaryotic material. The green dots are coming from the fish staining, so it's UBA DNA. And the red dots are actually a silver grain coming from telling us the activity of the cell or not. So you see on this picture that actually UBA has been found 2 times, but it's not active. And once we analyze the pictures, the software Action Vision gave us a file that was giving us the silver green area. We linked the silver green area to the leucine and DAC incorporation rate, and this gave us a regression line. We used this regression to convert this silver green area into activity that could give us the activity of the cell. The activity was pronounced in mole per cell per day, and for DAC, we convert it in femtmole per cell per day. So first of all, we can see the UBA relative abundance. As you can see, UBA has been found in the whole water column from the surface to the deep layers between 0 and 2%. We have 2 exceptions. In those cruise, you can see that in the AP and benzo-pelagic waters, UBA has been found from 2 to nearly 6%. And in Moby Dick Station 2, you see that actually the abundance of UBA was relatively low, between 0 and 1%. But except these 2 exceptions, we see the same pattern that UBA is more active, more present in the mesopelagic layers. And it's also present in the deep layers, as you can see here in Archimedes and in GeoTrace cruise. We also studied the activity of UBA. So among all UBA cells, which one were incorporating leucine and which one were incorporating DAC. On the left graph, you see leucine incorporating UBA among all UBA cells. So what we see is that among all UBA cells, between 25 and 80% of UBA cells are taking up leucine, which is really quite high. And you see there is a peak of activity in the upper bathypelagic layers. Moby Dick Station 2 has a little bit of different pattern because we see that here between 75 and 80% of the cells are incorporating leucine. But this could be explained by the environmental condition. Indeed, the Moby Dick Station 2 is a station with where there is a lot of primary production, and we suppose that this high primary production is the reason why UBA was way more active in this station. When you look at the graph on the right, you see that UBA is taking up DAC in less percentages, but still between 5 and 25% in the epi and upper mesopelagic layers. But you see that it's rapidly dropped to 0 with the depth. So when we look at the 2 graphs together, we see that kind of a pattern. So UBA fixing DIC in the upper layers and UBA incorporating leucine in the deep layers. On this slide, I wanted to show you the microscopy result that I get. So on the top right, you see pictures taken with the camera of the on the microscope. So you see DAPI channel, so it's only prokaryotic material. Then on the bottom right, you see the DAPI channel plus FITC channel, meaning a prokaryotic material and UBA stained cells. And on the left side, you see the 3 channel combined, so meaning with the translate channel from the autoradiography. And you see the dark points coming, actually, they are silver grains, so meaning they are indicating the activity of a cell. So as you see here, we see 1 UBA positive cell, a leucine positive cell here. And here we see an aggregate of different UBA cells incorporating DAC. We know it's an aggregate because of the size of the cell. It's too big to be UBA. And also when we look independently at each channel, we saw actually many UBA cells aggregating together. With these 2 histograms, you can see the single cell activity of UBA and the total prokaryotic community in the Northern Atlantic. So on the left side, it's leucine incorporating cells, and on the right side, DAC incorporating cells. On the left side, you see that actually the average activity of UBA is very close to the average activity of the total prokaryotic activity community, around 0.05 to 0.25 from the surface to the deep layers. The interesting point is that the activity of UBA and DAPI does not change that much with depth, and they're still active in deep layers, but the activity remains relatively low. And the fact that UBA and DAPI has the same activity prove that UBA actually has could have the same role than the total community in the carbon cycle. On the right side, you see DIC positive cells. It's the same constat. We see that actually DAPI and so the UBA stained cells and the DAPI stained cell, which is the total prokaryotic community, have the same activity, so from 0 to 0.25 femtmole per cell per day. in the surface layer and upper mesopelagic layers. And we see that under 400 meter, we see that there is no more UBA incorporating DIC, except this peak as 2000 meter. But we have to take this peak very carefully because, as you saw, here it's the total count of cell, meaning that if we only found one cell incorporating DIC at a rate of 1 to 1.5 femmole per cell per day, it can reach 100% immediately. So this has to be taken very cautiously. However, the trend was not observed in the Southern Ocean. Indeed, at station M2, you see that the average activity of UBA is from 0.4 to 0.8 atmol per cell per day, where the average activity of the prokaryotic community is more around 0.2, 0.4 atmol per cell per day. And this is observed along all sample depth. But again, as I said before, this could be explained because of the environmental condition, meaning that UBA, when there is a high primary production, is highly active and could take up a leucine very easily. Because on station M3, for example, which is very close to station M2, but with a very low primary production, you see that UBA main average activity is way more lower than the total prokaryotic activity, meaning that station M2 is very different from the rest of the stations. In this table, we can see the contribution of UBA to the total leucine incorporation and DIC fixation among total prokaryotic activity in percentage. And what we see kind of interesting, we see that actually the minimum value of UBA fixing a leucine is 0.3%, and the maximum is 3.8 in zone. And for the DIC fixation, we see that the minimum is 0.4 and the maximum is 2.8. And the fact that these figures are at the same range show us that actually UBA is not heterotrophic as we thought at the beginning, with some tendency to change its metabolisms. UBA is actually mixotrophic organism because of this rate that actually very similar. As a conclusion, we can say that UBA has been found in the global ocean through the whole water column, with a relatively high abundance in the mesopelagic waters. The metabolism of UBA has been determined as heterotrophic and autotrophic with DIC fixing, and the rates have shown that UBA was actually a mixotrophic organism. The shift of metabolism observed from the surface to the deep waters is also a point of interest, and this allows us to say that active UBA are contributing to the geochemical carbon cycling in the global ocean. Aggregates of UBA fixing DIC were found in the epipelagic waters and could be a next axis of research for the future. I want to thank, first of all, Professor Gerhard Helm, that was the one allowing me to go to Vienna to do my master thesis. I want to thank my supervisor, Chi Amano, which was the one helping me in the lab and also for the analysis. I want to thank Federico Baltar, because he was the one coming up with the topic, as well as Thomas Renthaler for the sample. I want to thank Murat Skisadur for the autoradiography help, and also the whole microbial oceanography group. And I want also to thank the IMBRC program as well as the Erasmus program that allowed me to do this master thesis in Austria and this great experience. Thank you very much for your attention. And if you have questions, please, I'm listening right now.